Okay, so continuing talking on about enzymes. Yesterday we talked about the active site, tertiary structured, which is those R groups in proteins. Their electrochemical properties forming and shaping enzymes so that they have a specifically shaped active site, so that they can the active site is attracted to the substrate. They can join with the substrate and do things. So here's my enzyme, and it is attracted to the pen, and oh, this pen doesn't have a cap. Here we go. My enzyme is attracted to the pen, and it breaks the pen apart. And so now the product is these two things. And the enzyme, the active site, is no longer attracted to these two things, so it lets them go. The enzyme can continue on and keep splitting the pen open. This enzyme would be called green penase. It could also put the two things together. Cap pen synthase. Sure. What else did we talk about? So it's like a lock and key. That's the first kind of model. And there's also induced fit, where these things come together and their electrochemical properties change the shape of the enzyme, which makes the enzyme work. And that is more likely the true to life. Apoenzyme is where it's just the protein bit, and you have a coenzyme that comes in, so that can be like a metal ion, which takes electrons, usually those clean up electrons from a reaction, or a coenzyme, which will be some kind of organic, a vitamin, some kind of, or it'll be another bit of protein. So, my other hand comes together, and now this enzyme is active. Okay. Anything I'm missing from there? I don't think so. So factors affecting the activity of enzymes. That's our next bit. So enzymes, we want to maintain homeostasis in or cells in our cells and in organisms. So homeostasis is a range of conditions that lets the cell live. So there would be temperature and pH mainly. So the reason that we want to keep homeostasis is so that the enzymes can work, because enzymes work in a range of pH and temperature. When the temperature starts to get too hot, what happens is those hydrogen bonds that are holding that tertiary structure together, when there's too much energy in the system, when it's getting too hot, they start to break apart. And the enzyme loses its shape. And shape is the important thing here. That shape, so that it perfectly, like lock and key or induced fit, fits over the substrate. So temperature. So when things get hotter, the enzyme starts to denature. Those H bonds start to break and it starts to denature. When you cool it down, those hydrogen bonds can form again. If you get it too hot and you start breaking beyond the H bonds, so the covalent bonds that are holding the enzyme together, or those ionic bonds, so that that tertiary structure and secondary structure start to come apart, the enzyme's not going to come back together. But if you just heat it up so that those H bonds break, that tertiary structure, you could deactivate the enzyme. But once things cool, it can come back together. And this is used, for example, in PCR, which is what BC is using to check if you have COVID-19. They do a PCR test, which is they take DNA basically and they amplify the DNA. And they're looking for specific parts of DNA. They use an enzyme, a DNA synthase, an enzyme that makes DNA, and in the process, they can turn it off and on again by heating up and cooling down the reactants. So when they don't want it on, they can cool down, and then when they don't want it on, they heat up the mixture, and then they can do other stuff, and then they cool it down to activate the enzyme, and it just goes off and on like that. Okay, the other thing is pH. Oh, the other thing is cooling enzymes down. So when you cool them down, they don't actually denature. When you cool enzymes down, remember enzymes raise the activation energy. or lower the activation energy. Sorry, it's a rough morning today. Lower the activation energy. So they bring the reactants together so that there doesn't have to be a huge amount of energy to slam them together and start a chemical reaction. The enzyme does that. The actual enzyme slams the things together, so it doesn't have to be so hot. But when you cool things down, the enzyme actually becomes kind of rigid, more rigid. So that induced fit hypothesis, when you think about it, it's harder for it to change shape and for it to move. Also, because it's getting cooler, because it's rigid, all of the molecules, everything is moving slower. So it might be harder for the enzyme to run into the substrate, because now the enzyme is moving less, the substrate is moving less. Everything is just slowing down until there's, you could get it so there's not enough energy to even bring these two together and for the enzyme to move and do its job. pH. So also, if you have too many hydroxides, too many of those hydrogen ions, they're going to attack those H bonds, especially when things get acidic. You're going to get those H plus ions are going to be, they're called hydrogen bonds. And if there's all these hydrogens around, they're going to start ripping up the enzyme and they're going to start tearing up those hydrogen bonds. So the enzyme will denature. So enzymes work in a range of pH depending on what the R groups are. So, and they can work in acidic conditions. For example, let's see, there's pepsin and trypsin. So they both are peptidases. So they break peptide bonds. Pepsin works in the stomach, very low pH. And trypsin works in the small intestine, which is basic, has like a pH of 8 something. So, and they both break peptide bonds. They both help you digest proteins because it's very important. So they might have the same active site, but the rest of the enzyme might be different and therefore better suited for basic conditions or better suited for acidic conditions. Okay, what else do we have? So hypothermia, hyperthermia. Hyperthermia is when body temperature rises above 37 degrees centigrade. So all the enzymes in our body are really, their temperature range is in that 37 degrees centigrade area, around there. And hyperthermia is when your body starts to get too hot. And you enter hyperthermia. So your body tries to maintain homeostasis, so it's trying to keep you at 37 degrees centigrade. So there will be feedback loops, which we'll talk about in a second, negative feedback loops that will bring you back down to 37 degrees centigrade. Hypothermia is when you get too cold, when your body temperature drops too low and there's not enough kinetic energy to carry out metabolic functions. So below 35 degrees centigrade, body enters hypothermia. And so, then what happens is you start to shiver and you get goosebumps and blood goes to the center of your body away from your extremities to warm your vital organs and to keep those enzymes running. Now, another thing affecting the function of enzymes is inhibitors. So you've got an active site that's tuned, that is specific for the substrate, but there can be other things, like this red pen maybe, so if I have this enzyme that is green penase, let's just say it's a synthase, so it's green pen synthase. It brings the cap and the pen together. And there's a red pen, and the red pen's shape is very similar to the two green pen, but it's not exact. It's an inhibitor. So what happens is it joins with this enzyme in the active site, and now the enzyme is trying to put this together, and a green pen might be coming along to get synthesized, but it can't because the active site's already filled with red pen. So the red pen is an inhibitor. It fits in the active site, and it's a competitive inhibitor. And it doesn't let the enzyme do its work. So a lot of medicines act this way. So antibiotics act this way. They interfere with bacterial enzymes so they can't do their job. A lot of drugs work like this. So drugs inhibit parts of the functions of your brain, your metabolism in your brain. There's you have happy neurochemicals that get released, and they activate receptors, and you feel good. You feel very happy. So there could be, and then what happens is then those happy chemicals get eaten up by an enzyme, and you stop feeling good again. Some drugs are an inhibitor to that enzyme that eats up the happy drug, and so what happens is you keep feeling good. Your body doesn't remove the happy drug. The problem, of course, is that screws up homeostasis. It screws up all the things. So that's a competitive inhibitor when it comes into the active site. A non-competitive inhibitor would be something. So here's the enzyme that synthesizes the pen together, but an inhibitor comes along. So I don't know if I stabbed my hand with this pen. So it doesn't affect the active site. It attaches somewhere else on the enzyme. It affects those electrochemical bonds. And so if I stabbed my hand, so I screwed up this hand, now this hand can't move, so the active site is twisted, is changed, and so it can't do its job. It can't form the enzyme substrate complex. Right? Remember the ES complex. Enzyme substrate complex is when the enzyme and the substrate come together and make the product. So if we affect that, so if we inhibit it in the active site, that's a competitive inhibitor. In the non-active site, it's a non-competitive inhibitor. And last but not least is heavy metal ions, so like lead. When you ingest heavy metal ions, these are the reason they're toxic. Because what they do is they have this huge positively charged nucleus, and so it rips hydrogen bonds apart. It changes the electrochemical properties of enzymes. It can bond to negative things and weigh them down, basically. So it's just this large positive nucleus. And remember, all of this works on these electrostatic forces. So this large positive nucleus disrupts those electrostatic forces. Okay, next thing that affects enzyme activity is substrate enzyme concentration. So if you have lots of enzyme, then the reaction rate will be really fast because you're going to encounter substrate all the time. You've got lots of enzyme. You keep adding more enzyme, you're gonna until all of the substrate is gone. And if we look at the graph of enzyme concentration, it's just a straight line. The more enzyme you have, the faster the reaction rate until it drops down to nothing. Now, if you add substrate and not enzyme, so if the substrate concentration goes up, eventually all of the enzyme molecules that you have, their active site will be taken up by a substrate. And so, everybody will be in an enzyme substrate complex. And any more substrate that you add will have to wait in a queue until the enzyme has completed the reaction, and it can move on to the next substrate. So the reaction rate will be limited to a maximum. So you're adding substrate, but eventually all the enzyme substrate complexes are full, and so you will just get a flat line coming across. This is substrate concentration versus reaction rate. Okay? So just be familiar with those two graphs and be familiar with how that works. When you add more substrate, you're going to saturate all the enzyme. When you add enzyme, you're not going to saturate all the enzyme. It'll just go faster and faster because more and more enzyme will encounter more and more substrate. Okay, we're supposed to do all of this on B4 until B5. So let's just continue on. This is going to be a longer video, I'm sorry. We're squeezed for time. Because tomorrow we have to start membranes. So, metabolic pathways. So a metabolic pathway is just, a big reaction. So we're going to use here an example of taking glucose and turning it into ATP. What happens in a metabolic pathway is that doesn't just happen in one complex go. It's broken up into little steps. So this happens, then this happens, then this happens, then this happens. The advantage of that is you have an enzyme for each one of those. You have a low activation energy for each one of those. You don't have to have some big jump. So it's easier also to maintain control this way. So you can, there can be inhibitors because you're trying to maintain this homeostasis so you don't have a runaway of making ATP or whatever it is that you need to do. You can control, you can inhibit. Sometimes there's feedback loops, so the products of one step will inhibit the previous step or will be a coenzyme for the next step. These kinds of things. So let's just talk about turning glucose into ATP. So the first step, so be familiar with these, so is called glycolysis. And glycolysis, every cell will do glycolysis. Eukaryotic or prokaryotic. Remember, eukaryotic cells are our cells. These are cells with membrane-bound organelles and nucleus. They have compartmentalization. Prokaryotic cells, no compartmentalization, no nucleus, everything is just in the cytoplasm. So prokaryotic and eukaryotic cells will do glycolysis, which is basically they take a glucose, turn it into fructose, then the fructose gets split into these two 3-carbon units. There's NAD, all kinds of stuff happens, and they get turned into pyruvate. And this produces 2 ATP. So basically, off this, you get 2 ATP. And remember, ATP is the energy currency of the cell. The P in ATP is the phosphate group. The bond, the TP is triphosphate. There's 3 phosphates. The bond that holds the third phosphate to the second phosphate is a high energy bond. When it's broken, it releases energy that the cell can use. And that energy is just heat. It just increases the kinetic energy of the area around it. So one molecule of glucose goes through this process and produces 2 ATP. In eukaryotes, then what happens is these pyruvates, they continue on in the presence of O2. So this is aerobic respiration. Here we don't need O2. It's anaerobic respiration. But in the presence of O2, they go into the mitochondria, and the mitochondria is the powerhouse of the cell. Because here, across this double membrane, there are lots of proteins here that carry out the Krebs cycle. And so there's a bunch of steps, and it produces 36 ATP, some CO2, and some water. So in eukaryotes, look, one glucose produces 36 ATP plus 2 ATP is 38 ATP. That's why eukaryotes are so much more efficient, because from one glucose, they make so much more energy. Whereas prokaryotes, they only make 2 ATP. If eukaryotic cells, if there's no oxygen, so this happens, your muscle cells can do this, they'd be what's called facultative anaerobes. So you're running, you're not breathing enough, and your systems cannot keep up. There's not enough oxygen getting to the muscles in your legs. What your muscles in your legs will do is they will just do this. And so they're still getting ATP, but just not as much. They're not as efficient, so they start burning glucose at a higher rate, and they're making pyruvate. And what they do is they turn this pyruvate into something called lactic acid. That's why your muscles start to hurt after a big workout. And then eventually when oxygen comes back into the system, that lactic acid is turned back into pyruvate and burned for ATP. It goes into the mitochondria to make those 36 ATP. In plants, in fruit, if there's no O2, they go into fermentation as well. This is called fermentation when you just do this bit and you're an animal cell or plant cell. But instead they make ethyl alcohol, which does not then be burned for when oxygen comes back into the system, it doesn't continue on to be in the mitochondria, but instead we usually harvest it and make liquors and spirits and wine and things like that. Okay, last but not least are these feedback systems for maintaining homeostasis. There's two kinds. So when you have these metabolic pathways, so this is just an example of one. So we're trying to maintain 37 degrees centigrade so the enzymes can work. If your temperature goes up, there's a feedback system. So you start to sweat, your blood goes to the extremities to cool down, and your temperature starts to drop. Okay, and that's a feedback system. If you start to get too cold, you start to shiver, blood goes to your center, and you start to heat up. So these are negative feedback systems. Basically, the product of one inhibits the previous step in a metabolic pathway. Positive feedback systems like this, this looks crazy. This is when I was doing these notes. This is way back when I was like drawing, and it's all somehow messed up. But so, for example, when a woman is pregnant and it's time for the baby, she has a contraction and a muscle contraction. When she has a muscle contraction, that releases a hormone called oxytocin, and oxytocin stimulates more contractions. More contractions stimulates more oxytocin. More oxytocin stimulates more contractions. More oxytocin, and on and on and on. It's a positive feedback loop. Whereas, so the product of one step accelerates the previous step, basically, which produces more product, which is a big circle until finally the baby comes. So, negative feedback maintains homeostasis. Positive feedback breaks homeostasis and reinforces a change until a new condition is met. Okay, that's what we have for today. Go through that, answer those concept questions. See you tomorrow.